Okay, so today we're going to talk about modern genetics. On Friday, we saw that how they discovered the shape of DNA, and we saw that it was a double helix, which is represented here. And what makes up DNA are these structures here. These are called nucleotides. And what makes up a nucleotide? Well, we have a phosphate group. We have a sugar, and this sugar is called deoxyribose. And we have a nitrogenous base. Now, what makes each what makes each nucleotide different are these nitrogenous bases. When we talk about DNA, we have four different possibilities. We have adenine, we have guanine, we have cytosine, and we have thymine. Okay, so like I said before, the nucleotides are all the same, have the same parts, but the bases differ between each nucleotide. There are only four different nucleotides. And here, if we look at this double helix, the outside structure of the double helix is made up of the phosphate groups and the sugars. The inside rungs are made up of the nitrogenous bases. And what makes this twist and form the double helix is the relationship or the structure of these sugars or nucleotides bonding together. They have to bend and twist in order for the bonds to match up. Okay, so if we go to the next page here, we can see that we have four structures, and each one of these represents one of the different nucleotides, and you can see the four bases are represented differently, and they kind of like puzzle pieces. And with okay, so you can see how each of the bases are different. I'm sorry if I lost my spot, but I had to pause it before because the bell rang, and so I may repeat myself, sorry. But these four shapes are different, representing the four different bases. Okay, so back to that video on Friday, we noticed, if you remember, they said that they learned from an experiment that there were equal numbers of adenine and thymine and equal numbers of guanine and cytosine. So that led them to believe that how these connected were that adenine and thymine always bond together. Right? So if we make this one here adenine, then what would happen here is this one would be thymine, and we'd have to turn it around and bond it and click it together. So this one would have to be thymine. And what's holding these two nucleotides together at the bases is a hydrogen bond. Now, this one here, if this one is guanine, then this one would have to be cytosine, and this is the match. And they would click together, also being held together by hydrogen bonds, and then they would be bonded this way. So they're running in opposite directions because they have to flip in order for the nitrogenous bases of each nucleotide to blend together. Okay, so that, and then let me put a C here for cytosine. So sometimes on the Regents exam, you'll see they'll draw it this way, and then they'll have adenine, guanine, cytosine, adenine, and just random bases, and they ask you for its complement. So you would have to know that A always goes with T. Oops. A always goes with T. G always goes with C. C always goes with G. A always T. T goes with A goes with T. C, and G, and then you would just complete it. So this would be the completed double strand or double helix. Now, when we talk about RNA, because we have two types of DNA that's or genetic material that's found in our cells, we have DNA and RNA. The 4 bases are different. Well, I should say one of the 4 bases are different. So what I want you to do is look up how RNA is different from DNA. All right, so the obvious one, right? So if DNA stands for deoxyribose, hold on, I'm sorry. Deoxyribonucleic acid, and the other genetic material in our cells is called RNA. So just looking at the name, can you figure out what one major difference is between the two? Right? So remember, the sugar for DNA is deoxyribose. RNA, right? What do you think the RNA is? What do you think is different? The sugar, the phosphate, or the base? Right? So deoxyribose was the sugar. This one has a different letter, so it's safe to assume that it's the sugar a sugar is different, right? So it's actually ribonucleic acid. So what I want you to do is figure out how the bases are different for DNA and RNA. And I'm going to give you a DNA structure here. So let's see, A, T, C, A, G, 1, 2, 3, 4, 5, 6, 7, 8, 9. I want you to make the RNA copy of this. If you could use the internet or your textbook. We're going to go over it in class on Monday, but I want you to see if you can figure out how to make the RNA complement to this. Right before, this was the DNA complement. So I want you to make the RNA complement of this, just using the letters like we did before with DNA, for class on Monday. Okay? So thank you for watching, and I'll see you Monday.